Hello again. If you're wondering why do I have to keep my videos short and I have to keep why don't I do long videos, and it's just simply because it doesn't the system doesn't allow for because if I go on and on and on, the files be very very huge, and it'll take time to render as well as it will take time to upload. So that's why I just keep it small so that it's easy for you to go through. All right, okay, now sorry, so we go on right to this experiment. Remember I mentioned in the previous video, it says we know that bacteria, for bacteria the DNA is genetic material, for viruses that has DNA is genetic material, but now we're talking about RNA viruses, in which, so what about RNA? Is RNA also a genetic material? And experiments here was done by Frankel, Conrad and Singer in 1955-7, somewhere around that time. What they were able to do is an experiment is called hybrid virus reconstitution. Now what they are able to do is they are able to take viruses apart and put them back together. All right, they can take viruses apart and then they separate the genetic content and the protein code, and then they were able, they have means and ways to actually put them together again. Now there are two viruses. For the sake of clarity, I'll just use this one. It just says type B and type A. Actually, it's if I'm not mistaken, the Holmes ribgrass variety. But never mind, we'll just call them TMV. TMV is tobacco mosaic virus, so type A and type B. What they do is they degrade, right? They degrade, as I said, they just separate the protein code from the DNA, and they took one code, they took a code from one type and then the RNA from another type, and then what they did is a hybrid virus. Now they made several hybrids. They have they made type A code with type B RNA. They have type B code with type A RNA, and also they made the original ones, and then they infect plants. This is remember, it's actually tobacco mosaic virus, so actually attacks tobacco leaves. And from when we also know that if viruses attack, that means it's going to make more of its own, more viruses, correct? So after infecting the bacteria, they isolated the viruses from there, and the conclusion was it's like the acetobularia experiment. It depends on the genetic, it depends on the RNA type. Regardless of the protein type, if the RNA type is A, even if the code type is A, or even if the code type is B, you will still get an RNA-A virus. You do not get hybrids. If the type of the RNA is B type, you will get a B virus, a complete virus. So again, this is an elegant experiment to show that RNA is a genetic material. RNA is the material that determines what type of RNA and protein that is going to be made in the coming generations. All right. So 3 conclusions from the previous video. For bacteria is the DNA genetic material, DNA viruses, DNA genetic material, RNA viruses, RNA is genetic material. So basically all this say that between protein and nucleic acid is the genetic material. All right, so here you go, nucleic acid is the genetic material now. Okay, so in the next few other lectures we will go on to look at maybe I could just continue in this slide. Now for nucleic acid structure and function, I think I have videos that I've actually selected from YouTube, which I think is quite relevant. But there are some genetic characteristics that I can actually explain here. Now DNA is a polymer, is a chain. All right. and this chain is made up of nucleotides. All right, I think we all know that. Now what you probably may not know, this is a nucleotide. All right, a nucleotide is also called a nucleotide monophosphate. All right, a nucleotide is a specific name that is used for a molecule that has one base, one sugar, and one phosphate group, mono. So because if you know that you have ATP, adenosine triphosphate, correct? So ATP is a triphosphate, so it's not a nucleotide, right? It's a mononucleotide. Sorry, it's a deoxynucleoside triphosphate. Sorry, nucleoside is this two, and monophosphate, one phosphate. Nucleoside triphosphate. Yeah, so you need to just, I think there's later on there's a table that describes this, but never mind. So basically you need to understand that if you say nucleotide, you're only talking about one base, one sugar, and one phosphate, and this is what makes the DNA molecule. There are other types of nucleoside diphosphates, nucleoside triphosphates. Those are actually not called nucleotides. They are made up of two types, purine and pyrimidines. You need to remember them. From my student days, I remember them as in my student days in Malay it's called pyrimidina and then purina. So this is a Malay word for it. So I used that and made a girl's name, Siti Pyrimidina. All right, so that means C and T is actually pyrimidines. If that works for you, you may have your own way of remembering. So Siti pyrimidines and AG are purines. Now a DNA molecule has a direction. It is, you know the Lego blocks, they can only go one way, right? Lego blocks do not, well, of course you can, one way meaning you have a top and you have a bottom. So similarly you have that characteristic in the DNA. You have a 5' end, you have a 3' end. If you look at this closely, you will find that the 5' end, it depends on the C, the carbon. Now if you go back one slide here, okay, you look at, can you see the numbers? If I enlarge that a bit, okay, this is how it is being numbered. Remember, you have the number starts here, not here. Remember, when you say 5 carbon is a pentose, the 5th carbon is actually on the outside of the ring. All right, so you have carbon number 1, carbon number 2, carbon 3, carbon 4, and carbon 5. Yeah, it's always the same numbers, the same orientation, and the bases are actually connected in carbon number 1. The phosphate is at carbon number 5. The OH group, the hydroxyl group, is in carbon number 3. It's the same thing with purines. All right, you have again, you have the 1, 2, 3, 4, the carbon number 5 on the outside where the phosphate is located. Carbon number 1 is where the bases are, and carbon number 3 is where later on we'll see that it's important for polymerization. Okay, so now here you can see again that's carbon number 5, and the bottom carbon number 3. So this one is connected to phosphate to carbon number 5, and then carbon number 3, phosphate. So it's always 5' and 3'. All right, so there's a direction. So when you read DNA, normally you will read 5', 3'. Like the your books, for example, you will read whatever text you write is from the left to the right, whereas in Arabic it goes from the right to left. For DNA, is always reading 5' to 3'. Doesn't matter how you write it. So that's important that every time you write DNA, you must have the 5' and 3'. A DNA sequence that does not have 5', 3' cannot be assumed to be reading one way or another. All right, so every time you write DNA, you must have the 5' and 3' end. Okay. There are two types. Now look here, what's different here. You have one is DNA, the other one is RNA. This is the deoxyribose. You have the ribose, What's important here is the absence. Here you have both OHs, and here you have only one. That later determines whether this can be used for elongation. It has probably on not this stage is that not important for you to understand, but you need to know the difference between ribose and a deoxyribose. This one makes RNA, this one makes DNA. Okay, DNA structure and properties. I'll continue the next slide. Bye bye.